see, according to the Pasteur, virus is a
Venom.
Latin word, and yes, virus is venom or poisonous fluid or any type of the harmful secretion. Okay, that point you remember. Ivanovsky, contribution of Ivanovsky is given there. Separated the microorganism TMB. Mosaic, this is in the tobacco plant, this is known as tobacco mosaic virus. And Beijing demonstrate that from the TMB, the infected material is isolated, is known as contagion fluida. Virus is submicroscopic and non-cellular, smaller than. Now it is not given. Virus submicroscopic non-cellular smaller than 200 millimicron or 200 nanometer, both are same. Yes, millimicron or nanometer, both are same. Milli, 10 raised to minus 3, micro 10 raised to minus 6, so total 10 raised to minus 9. Clear? So nanometer is also known as millimicron. Now, virus is in outside of the host body. But when it is entered into the host body, become active. But yes, that's why it is connecting link between living and non-living organism. That you know. But after entry in the host cell, the host cell consists the ribosome. So host cell's ribosome used as a protein manufacturing factory by this genomic virus. Okay, so the gene of the virus, you know that virus in the genetic material is RNA or DNA, but not both. Virus consists genetic material as a RNA or DNA, but not a both. And listen, RNA is in the form of SS or DS, and DNA is also in the form of SS or DS, but both are not present there. We have both. We have both. Fine. So for metabolic process, we have RNA. 3 types of RNA we have: rRNA, tRNA, and mRNA. Okay. So host cell's ribosome can be used by the genetic material of the virus, and according to the virus gene, our ribosome forms the protein according to that codons present on the gene. Okay, one thing, actually mostly any type of the metabolic activity, just I can say you like that regulation of gene expression. એક વર્ડ તમને આજે નવું આપું છું. What I told you?
Regulation of gene expression.
Gene expression. Gene can be expressed its characteristic by formation of the protein from it. So here the sequence is that DNA is transformed into mRNA.
Yes.
Along with that mRNA, rRNA, tRNA are also synthesized there. And with the help of these three RNA, M, T and R, on the ribosome, mRNA is situated there, and over the codons on the mRNA, according to that cause, protein is synthesized. I will show you the one video in 12th standard at the time of the molecular basis of inheritance, 6th chapter, how the DNA is transcripted, how the mRNA is synthesized, and how the protein is synthesized on the mRNA in the ribosome. Can you got this each and every word?
Yes.
So DNA is converted into RNA, RNA is converted into protein. This whole phenomenon is known as regulation of gene expression. Actually, we say like that each and every organism is different according to their protein structures. આપણે એવું કહીએ છીએ કે બધા જ ઓર્ગેનિઝમ ડિફરન્ટ છે because they have different proteins in their body. Yes, but indirectly we say like that each and every organism has different DNA. DNA code is different, RNA code is different, protein is different. Okay, so this is known as regulation of gene expression. તો અહીંયા કેવું કરે છે કે વાયરસ તો એક તો પેરાસાઇટ છે, બરોબર, and it enter inside our body. Then the genetic material present in the RNA, that is poisonous one, it affects the cell which is situated inside our body. It converts the genetic material according to it of our cell. So our cell is infected by the virus, and now the virus infected cell can perform the activity according to the genomic material present inside it, but use the ribosomes which are present inside the host cell. Okay, so please understand this. Tobacco mosaic virus is TMV 300 millimicron into 220 millimicron. Or we say that 300 nanometer into 20 nanometer size. spherical in shape influenza virus. Actually, the full name is H. influenzae, Haemophilus influenzae. It causes flu. It causes flu, and along with the Haemophilus influenzae infection and the Streptococcus pneumoniae bacteria, both are combined together and cause the severe pneumonia in human being. Okay, so influenza virus, HIV, human immunodeficiency virus. This name indicates the virus can depreciate the immunity power of the human being. So HIV mostly target our immune system. It kills the helper T cells and reduce the immunity power. As a result, person immunity power is very low. The person is unable to fight against a particular disease. The HIV affected person can go to each and every disease but cannot cure at all. Okay? Just think about it. If we have any type of disease, then our immunity power is affected. Then and then we got the disease. So generally what happens, if any type of infection inside our body, so gradually with the help of these drugs, antiviral drugs, antibacterial drugs, antifungal drugs, etc., with the help of that particular drug, it kills the affected organism, and then gradually our immunity power increases. And then we are able to cure against that. Then we say that we are normal. Right? But here, immunity power is decreased in such a way. Generally what happens, HIV છે એ બોડીમાં એટલું બધું ઇન્ક્રીઝ થતા જાય છે, એની મલ્ટિપલ કોપીઝ ફોર્મ કરે છે, and gradually whole immunity power ને decrease કરી દે છે. And that's why the reason is this. There's no any type of the vaccine, no any medicine, no any type of the treatment is available for the AIDS. Yes, in first stage, it is curable. Because you know that early diagnosis, better cure. But if the infection is more, then it is very difficult to cure it. બરાબર, પછી સર્વાઈવલ ચાન્સીસ બહુ લેસ થઈ જાય છે. અને ફાઇનલ સ્ટેજમાં તો એને સ્કિન કેન્સર પણ થાય છે. યસ, એડ્સમાં સ્કિન કેન્સર, બ્લડ વેસલ કેન્સર, લિમ્ફેટિક ગ્લેન્ડ્સ કેન્સર, લિમ્ફ નોડ કેન્સર, બરાબર. અને આ બધા એવા ડિઝીઝ છે કે ઓલરેડી ઓર્ડિનરી સિમ્પલ ડિઝીઝની સામે બી એ ફાઇટ નથી કરી શકતા. તો આ બધા જે લિથલ ડિઝીઝ, ઇન્ફેક્શિયસ ડિઝીઝ, ક્રોનિક ડિઝીઝ છે એની સામે પર્સન ઇઝ અનેબલ ટુ ફાઇટ અગેન્સ્ટ, એન્ડ ધેટ્સ વાય ધેર ઇટ ઇઝ હાઈ. એન્ડ મેજોરિટી HIV જે છે એ મોસ્ટલી અત્યારે આઈવી ડ્રગ્સથી બહુ જ સ્પ્રેડ થઈ ગયો છે ઇન ઇન્ડિયા. મેજોરિટી હમણાં મેં તમને એક વાત કહી ને કે એડોલેસન્સ એન્ડ ડ્રગ એન્ડ આલ્કોહોલ એબ્યુઝ. Majority adolescence age, it is from the 12 to 18 years. Okay, and those individuals can take the IV drugs directly. Fine. And at the time of taking the IV drugs, they use the contaminated syringe as well as needle. Okay, they can take this contaminated needle as well as syringe from the garbage. And that's why hospital waste management is much more important nowadays. જે અફેક્ટેડ પેશન્ટ માટે જે સિરિંજ કે નીડલ યુઝ થઈ ગઈ હોય એને ક્યાંય થ્રુ નથી કરાતી, બરાબર. એટલે એ ડાયરેક્ટ આઈવી ડ્રગ્સ, તમને ખબર તો હેરોઇન, મોર્ફિન, કોડિન, બરાબર, આ બધા આઈવી ડ્રગ્સ. એન્ડ ઇફ ધીસ નીડલ જનરલી the disease is spread mostly by contaminated needle. Please understand this word, contaminated needle by HIV. That and that only used by the normal person. Okay, so along with taking the IV drug, they can take the HIV virus also inside their body, and gradually they are affected. So nowadays in India it is very common due to this, right? So it is spherical one, rose-shaped tobacco mosaic virus and tadpole-shaped bacteriophage. Actually, bacteriophage, you know that virus infected bacteria, okay, or it is known as bacterial virus. And here in the bacteriophage, bacteria cannot survive. Bacteria will die. Fine. Now, in plant virus, this line is given that in plant virus, mostly ssRNA present. One line it is given there. But in cauliflower mosaic virus, mostly in the plant virus, ssRNA is there, but in cauliflower mosaic virus, dsDNA present. Cauliflower mosaic virus in that DNA is present in a double-stranded form. Gemini virus or plant virus, curly top virus, ssDNA is present in as a genetic material. In tobacco mosaic virus, ssRNA is present as a genetic material. 2130 capsomeres are present in capsid and 5% RNA and 95% protein. Now, the bacteriophage virus. Yes, bacteriophage virus. Now listen carefully. The virus which affect the bacteria So virus which affect the bacteria known as bacteriophage virus. Generally dsDNA as a genetic material bacteriophage is mostly used in genetics. Bacteriophage. QB bacteriophage. This is the name of the bacteriophage. QB. In that ssRNA is there, and phi 174 in that ssDNA present. Phi into 174, this is the name of the one bacteriophage in the ssDNA present. I think you are confused behind the bacteriophage. Majority nowadays, we have modified the genetic material of the bacteriophage, and that bacteriophage we can use as a genetic level to enter the desired genetic material, that is DNA or RNA, inside the host cell. Okay, so bacteriophage infect the bacteria. Bacteriophage virus infect the bacteria. We can modify it. Listen carefully. We can modify the genetic material of the bacteriophage, which is now used in the genetic engineering in biotechnology experiment. You want any type of the introduction. You want any type of the introduction of that, any type of the foreign DNA, which consists the desired character inside the host cell. We have to use the bacteriophage. Majority, the whole biotechnology as well as genetic engineering, it is work mostly based on the E. coli bacteria. So E. coli bacteria are mostly affected by this bacteriophage, and we have to conclude that what happened inside it. અને કંઈ વાંધો નહીં, હજી જો ખ્યાલ ન આવતો હોય તો તમે એમ કહો છો કે DNA is genetic material. મેં તમને આગળ પણ કીધું છે કે what is genetic material, એ શું છે, એનામાં કઈ કેરેક્ટરિસ્ટિક છે, which are the properties are present in this material that we say that DNA is genetic material, RNA is not. Yes, RNA is genetic material in virus. Mostly RNA is genetic material in most of the virus, but in all the organism over the world. Fine? It consists genetic material as a DNA. Why DNA is known as genetic material? What do you mean by this? તો એના માટે આપણે બેક્ટેરિયોફેજ યુઝ કરીએ છીએ અને આપણે એ કન્ક્લુઝન આપીએ છીએ that DNA is able to enter inside the cell, protein is not. આપણે કોઈ પણ સેલની અંદર DNA એન્ટર કરાવી શકીએ છીએ. જે ડિઝાયર કેરેક્ટર વાળું DNA આપણે એન્ટર કરાવવું હોય એ આપણે કરી શકીએ છીએ. અને એની ઉપરથી જ ક્વેશ્ચન આવે છે કે DNA ઇઝ અ જેનેટિક મટીરિયલ. બરોબર, એની પ્રોપર્ટીઝ છે, એના ક્રાઇટેરિયાઝ છે. જો એ મટીરિયલથી જ ફૂલફિલ થતા હોય ક્રાઇટેરિયા, then and then we say that this is genetic material, otherwise not. Okay, so These are used in the genetics, genetic engineering. અત્યારે બી ગ્રુપ વાળા માટે genetic engineering, forensic science, farm D, doctor B farm M farm नहीं, farm D કહું છું. Doctorate in pharmacy. B farm M farm करता है superior degree. And farm D is mostly related to the forensic science. And you are very, very near. If you can do the farm D, then you are very, very nearer to the scientist. Okay? એટલે અત્યારે આનો ક્રેઝ બહુ વધારે છે compared to the medical and dental, because એમાં મેરિટ હાઈ જાય છે. અને આપણે બે એન્જિનિયરિંગ કરી શકીએ. આપણે 2 types engineering કરી શકીએ, genetic engineering and biomedical engineering. Biomedical engineering means the engineering in which the instruments which are used in the hospital, we have to manufacture it and sell it. Okay? The engineer for the manufacturing the hospital instruments. તમે હોસ્પિટલમાં જેટલા ઇન્સ્ટ્રુમેન્ટ જોવો છો તે કોઈ ઓર્ડિનરી એન્જિનિયર નથી હોતો, બાયોમેડિકલ એન્જિનિયરિંગ કહેવાય. બરાબર? એ આપણે બી ગ્રુપ વાળાને થાય જેનેટિક અને બાયોમેડિકલ. બાકી આ સિવિલ ને એ બધું ન હોય, મેકેનિકલ ને સિવિલ એ ગ્રુપમાં થાય. Okay, now sign of age. How can you define it? The virus which affects the cyanobacteria infect the cyanobacteria or blue-green algae. It is known as cyanophage. Then mycophage. The virus which infect the fungi known as mycophage, and in this mycophage dsDNA is present. Mycophage, in that dsDNA is present. Now, prions. Or we say that's flu virus. smallest proteinaceous infecting agent two diseases are given there. BSE, bovine spongiform, encephalopathy, yes, CJD. Okay, તે તમને ફૂલ નેમ લખાવું છું, આપ્યું નથી આઈ ક્રુઝફેલ્ડ જેકોબ ડિઝીઝ નહીં આપ્યું. CR જેકોબ આપ્યું છે ને એ નહીં, એનું CJD નું એ ફૂલ ફોર્મ, CR જેકોબ નું ફૂલ ફોર્મ એ નથી. હા એ કહીશું. Encephalopathy. See, encephalopathy is mostly used for the brain. Cephalo, yes. અને આપણે હ્યુમન માં ઘણા બધા ડિસીઝ થાય છે. Encephalitis. All the type of infection in the brain known as encephalitis related to brain. Fine. So mad cow disease in cattle and CR Jacob disease CJD. તો એનું ફૂલ ફોર્મ છે Creutzfeldt, CREUTZ, CREUTZ, FELDT. પછી તમને એક ઓલું ખબર છે alfalfa? Have you ever heard alfalfa? One type of grass. No, alpha monday. Sama deva alpha. अल्फा बीटा पार्टिकल નથી આમાં. અલ્ફા અલ્ફા ને ગ્રાસ છે. Prions is over. Alpha mosaic virus, which was the infection in the alfalfa, in the ssRNA is present. Okay, now here I can stop now.